Hello, in this presentation, I would like to share with you some of the ongoing research in the Pierce group, which focuses on the development of mechanistic models for metalloenzymes. So one of the projects we work on is essentially looking at a series of iron-dependent enzymes that are involved in the oxidation of sulfur in biological systems. So for example, if I look at a eukaryotic sulfur metabolic pathway, we can see that cysteine, which is a sulfur-bearing amino acid, sits in a rather central position. Okay, so these enzymes that we're interested in catalyze the oxidation of cysteine to form compounds such as cysteine sulfonic acid, hypotaurine, and taurine. And these are essentially stimulatory neurotransmitters that are also involved in maintaining tonicity within skeletal muscle. So they're crucial for both neurological and cardiac function. Now, as you can see, once we oxidize this particular reactions here, these are essentially a non-reversible process. So once we oxidize it to form these compounds, ultimately these are going to be excreted as sulfate. And so they come at the expense of all of these reversible processes involved in essentially methylation of DNA, redox buffering, and anabolic synthesis of cofactors and coenzymes. Okay, so where our interest lies in this is that if you look at a series of patients suffering from neurodegenerative diseases, what you find is there's an imbalance in sulfur metabolism. Now, this doesn't necessarily mean that it's the cause of these diseases, but it is certainly a biomarker of them. And so what we're trying to understand is, well, how this comes about. Obviously, you start to see a lot of oxidation of sulfur occurring in oxidative stress as well. And so this ties into a variety of just trying to understand how better these enzymes function that catalyze the series of these reactions of taking a thiol to form a sulfonic acid. And as sort of alluded to here, there's a whole class of these enzymes that just change in terms of what their overall substrate is. So drilling down on these specific enzymes. One thing that's really interesting is that regardless of what the substrate is they catalyze, what you see is that largely the makeup of the active site is conserved. You have effectively a mononuclear iron site. Typically, the iron is bound in a ferrous state or an iron 2 state. And then you have a series of conserved outer sphere residues, in this case, let's say a tyrosine, a histidine, and a serine, which we collectively call the SHY motif. But these are, regardless of what phylogenetic domain they come from, these two features are always conserved. Now, in the case of cysteine dioxygenase from mammals, we can see that when this binds to the iron site, it coordinates via a sulfur atom from the thiol and a neutral amine from essentially the amino group here. And we see some outer sphere interactions with the carboxyl group occurring out here. Okay. Now, if we're looking at, let's say, 3-mercaptopropionic acid, in this case from a bacteria, we see that we have a carboxyl group, no amine, and the thiol, but also sort of a bidentate coordination. Now, what's interesting is, even though these are changing, the substrates are changing in terms of kind of size and charge, again, the first coordination sphere at the metal is largely conserved, and the contributing outer sphere interactions are conserved as well. Now, we can identify some of these key residues, and we can sort of prove that this, let's say, for example, this histidine, which is present in both, is relevant to catalysis just by doing some simple cytodirect immunogenesis. So, for example, we can change that histidine residue to an asparagine. This sort of disrupts the hydrogen, or maintains the hydrogen bond domain, but it gets rid of the ability to ionize that particular proton. Okay, and so what we can see is we go from a curve that looks like this, where we have 2 ionizable groups in a pH regime, to something that has 1, and that means that this residue here is coming from our histidine residue. Similarly, we can basically change this tyrosine such that it can't hydrogen bond to the active site anymore, to the iron site, turn it to a phenylanilene, and what we see is both ionization groups go away. Okay, so we can establish that in native catalysis, these ionizations are relevant and catalytically essential. Okay, so if we break this down and focus on separate entities, like let's look at the contributions specifically from the iron site and the contributions from the SHY, or the hydrogen bonding site. So under normal conditions, all of these enzymes would have what's called an obligate ordered addition, meaning that the enzyme will stabilize it in the iron 2 or ferrous state, and it is completely unreactive to oxygen until it coordinates the substrate. So we have a bidentate coordination of our substrate. only at this point in time, once we form the enzyme substrate complex, is the enzyme reactive to oxygen, generating our sulfinic acid product here. Okay? So we can take advantage of this to do what's called a chemical rescue type approach. Okay, so this is a way to get at what are the types of intermediates essentially involved in this chemical transformation. So in our case, what we do is we take our enzyme that we purify right here. We intentionally use a chemical oxidant to take this from a catalytically active enzyme to a catalytically dead enzyme, the iron 3 state. And then when we bind our substrate to form the enzyme substrate complex, we have a really nice charge transfer species that we can observe. But again, it's static, it stays there. We can look at this using another technique that's similar to NMR called electron paramagnetic resonance. And this is useful for looking at systems that have paramagnetic ions and looking for types of interactions around the active site. For example, magnetic nuclei around there. So we can use both techniques to characterize this starting point. Okay. Now, in principle, if we can introduce an oxidant that can accept the next 3 electrons, okay, so we've already oxidized it 1 electron from iron 2 to iron 3, and if we introduce something like superoxide, we can generate an iron oxo species and in theory put this back on a catalytic trajectory to generate our product, and our enzyme should be active again at this point in time. Okay, so in doing so, we go from this state here, which we can characterize. We inject potassium superoxide, and you can do this either enzymatically or chemically. But what you generate is a transient species here with absorption features that we can see in the UV vis. And we can follow those as they decay. And we can see that as they decay, we indeed see formation of a product. Moreover, throughout the course of this reaction, we can trap this and look at this in electron paramagnetic resonance, which allows us to see how many unpaired electrons there are and how this behaves. And what we can find is that this particular species is essentially electronically equivalent to an iron 3 superoxide. So, meaning that this particular intermediate is a catalytically relevant intermediate on the formation to product. Okay, so this allows us to basically go through and say that the first step in this particular reaction is the formation of an iron 3 superoxide, and characterize this by UV vis, EPR, and in this case here we're showing some Raman spectroscopy to show the superoxide stretch. So now if we switch gears and try to focus a little bit on what is the function of this hydrogen bonding motif, so this SHY motif, which is in the outer sphere, it's not directly coordinated to the iron site, and we can see that it has a profound effect on what is called the oxygen coupling. Okay, so if you think about the overall reaction that it's catalyzing, we have our cysteine, we introduce oxygen, and then essentially in a perfect world, there should be a 1-to-1 molar ratio of 1 oxygen consumed for every product generated. Now, in reality, this never really happens. So what we can do is we can measure the rate of oxygen consumed here. We do the exact same thing by measuring the rate of product formation here. Okay, and what you can see is that if we look at kind of the asymptotic levels, these aren't the same value. It's roughly 73-74% coupled, meaning that we're consuming for every mole of oxygen, we're generating about 70.73 moles of product. Okay, and that's what you get with a wild type enzyme. Okay, if you switch around the symmetry of this L-cysteine to a D-cysteine, you're going to break its interaction with this tyrosine motif. And in doing so, what you can see is that oxygen is consumed at the exact same rate, roughly equivalent rate. However, formation of product is at roughly less than 10% of the consumption of oxygen. So you've completely abolished the fidelity of this enzyme. It will consume oxygen, but it's likely just spitting it out in the form of superoxide, hydrogen peroxide, or reactive oxygen species. Okay, so it's telling us that this hydrogen bonding motif is directly involved with the essentially creating the oxygen binding pocket for this enzyme. And we can test this a second way by using essentially a spectroscopic probe for oxygen, where we take nitric oxide instead of oxygen and bind it to our enzyme substrate complex, essentially this species here. And so if we look at our wild type enzyme, this EPR signal is very indicative of that. We see this here, and this basically represents stoichiometric binding of nitric oxide to our enzyme substrate complex. If we go to the same variants we looked at in steady state catalysis, I can essentially maintain the same hydrogen bonding motif, but I get rid of its ability to be ionized by adding an asparagine there. And you can see that the intensity of this signal drops down pretty dramatically, about 5-fold or so. If I get rid of its ability to hydrogen bond whatsoever by adding a phenylalanine here, this goes down even further. In fact, I've scaled this signal by about 5-fold, so it translates to about a 20-fold overall reduction in its ability to bind nitric oxide. And by extension, that means that it would also be the same way with oxygen. So you can see that this hydrogen bonding motif is essentially creating the oxygen binding pocket. It's what gives rise to its specificity for oxygen. So what types of work do we do in my group? Okay, so as you can imagine, it's a very interdisciplinary group. We have some students that come in are very interested in working on more of the molecular biology, biochemistry, and enzymology. And this would be very traditional sense. We go through, we manipulate the plasmid DNA, we can express this in E. coli, we can do various DNA manipulations, whether it's through PCR or mutagenesis or any sort of standard biochemical technique. We then express this in protein and then go through standard protein purifications to generate our isolated enzyme. One thing that is a little bit unique is we typically will grow these up in large volume fermentation vessels, so 10 liters at a time, so that you're getting an awful lot of cell paste and proteins that we can characterize spectroscopically further on. And so the idea is we go through, we purify our systems, we could do standard steady state, pre-steady state kinetics, solvent kinetic isotopes, and these sort of classic systems to look at the overall function of the enzyme. We can then drill down on this much more detail using spectroscopic techniques. Our lab is essentially experts in this electron paramagnetic resonance, and we can use this to look at interactions of, let's say, the electrons within the active site, which tell us the oxidation state. They tell us if something's binding to that particular metal. And then we can use pulse techniques, just like you would do in an NMR, to get structural information, to essentially see what are magnetic nuclei that are close to that system here, and we can extrapolate this to models to explain, okay, what geometry is going on here. Now, sort of behind the curtain in all of this is we do a healthy amount of bioanalytical chemistry. Obviously, in order to characterize these precursors, whether it's this particular project with sulfur oxidation or our other project that works on tRNA modifications, we have to do a lot of HPLC, UV-Vis, LCMS, LCMS-MS, and these are basically to characterize and most importantly, quantitate the amount of products being generated. Members of my group have gone on to do a variety of things. So, for example, I've graduated around 6 PhDs, a couple of master's students, and you can see that, for example, Phil Palacios, who just graduated, he's doing his postdoc at Carnegie Mellon University. Sinjani Sadar is a staff scientist in Atlanta at a kind of a bioanalytical facility. Josh Crowell, who graduated in 2015, is a faculty member at the University of Texas at Arlington. He's essentially a lecture track faculty member there. Catherine Vandekar is also a lecture track faculty at Tarleton State University. So there's kind of a broad distribution of some people going off to industrial work and some people going off to PhD work in that regard. I feel like there's a lot of opportunities for good training that will translate to ultimately high-paying jobs, hopefully. Okay, well, with that, I'd like to thank you for your attention. And again, if you have any questions about this type of work we do or have any interest, please feel free to reach out to me via email or just call me in my office or, stop by at some point in time in Science Hall or Shelby Hall, 3097D. All right, thank you very much.